interested in variation, sequence variation, that is genetic variation. And my main interest is in mutations, primary mutations that are basic DNA and amino acid base changes. So these are the easiest ones to analyze. And they were the ones that we began working on, almost 50 years ago in the field of molecular evolution. I have only been in the field for 35 years or so. So whether the mutations have arisen only months or days ago, or years ago, or generations ago, or thousands of years ago, and over millions of years ago, this is where I started my research, the millions of years ago, that I am very interested in them because the dynamics of mutations, their birth, their death, their fixation, their polymorphism, happens at different timescales. And all of these timescales can be analyzed using current methods and technologies, techniques and theories in molecular evolution. So I'm very interested in all of them. And today I'm going to try to cram in a whole bunch of different things. I won't talk about all of these topics because there's just too many, but I'll talk about a few topics that relate with mega and others. So my interest is in molecular data-driven discovery. So essentially, I'm interested in developing methods and algorithms because by training, I am a computational scientist or scholar. I'm interested in building software and databases that make these and many other methods we have available to everyone who wishes to use them, making it accessible. I'm interested in actually taking large amounts of data and deriving patterns from them. In fact, this is one of the major impetus for my research for developing new methods, because every time we try to use data, we realize current methods are too slow or too difficult to use or have too many assumptions. So we try to innovate. And ultimately, we are very interested in building models that can translate evolutionary knowledge into actionable information. So today I'm going to try to touch upon all of these things, but it might be a little too much. So I might stop at some logical point. But I'm interested in sort of giving you a spectrum of research we do. So essentially, since we do analysis of mutations, it is natural to do evolutionary computation, which is comparative sequence analysis. And this picture here shows you that very many people are doing it. And they've been doing it for a long time, but recently there has been a huge escalation. So on the right side, you see a picture in which you have more than 60,000 new research articles that have cited many software packages like Mega software in our field. So this is the impact of the field of molecular phylogenetics and evolution on general biology, where people are studying origin of genes, relationships of organisms, pressure that are selective in nature, biodiversity, and even genotype-phenotype connection using comparative sequence analysis. And this is what I feel is really exciting, that we are connecting with so much community on this topic or this kind of research. Now, the thing is, when you do evolutionary computation, you leave a big environmental footprint. Being a biologist, we are very interested in this problem. And of course, whenever we leave an environmental footprint, which is because we have to make energy, ultimately, which leads to carbon dioxide emissions, this deteriorates our environment. And also, when things take a long time to compute, then they just take a long time to compute. So we cannot do as much research and things are slowed down. So I have a lot of interest in that problem. And here is that the mega users also leave a lot of carbon footprint because mega contains many methods that can take time. But in fact, the number of users using mega, the number of researchers is very large. So if you multiply even small amount of usage per user, this is a huge usage. So this gives you an idea of this increasing trend of usage. And so this is the focus I have in the first part of the presentation is on building computationally efficient and accurate. So here, the idea is to develop approaches and methods that are not only efficient, computationally means they take less time and less memory, less computer power, but they also produce accurate results. So So this is more like you have your cake and you eat it also at the same time. And so we've been involved in this kind of research for many years, and I'm going to tell you a little bit about that on what we are doing for Mega. So in Mega 12, we have uh, come into adaptive and green computing, which is more sustainable, which is more exciting and faster for researchers. So it's good for the environment and it's good for research both. And so this version was just released in December. And for those of you who have used Mega in the past, you know, all our symbols used to be red. And I guess I knew they were red computing or creating bad for the environment. Now many of these icons are green. So this indicates that we are very interested in green computing. So, so essentially, I'll talk about a few green computing advances in Mega that may be of interest to you. So one is the model selection. In every phylogenetic analysis that we do, we sort of have a protocol. We do a model selection followed by phylogeny inference, followed by, in many cases, bootstrapping to test the phylogeny's robustness, and in some cases also molecular dating for the species or duplicate gene evolution. So I'll talk about some of these topics now about how we are helping with green computing and accurate computing for these. So substitution model selection, I will not discuss this too much, these models, but there are a large number of models, and in Mega you can test many of these. So substitution model selection on the x-axis here, you see, it is a time-consuming process. The time taken increases with the data size. More sequences and sites you have, longer it takes. It's a very straightforward thing for maximum likelihood. Total amount of data dictates the amount of time taken. And it's a beautiful linear relationship. So in Mega 12, one of the things we innovated to avoid analysis of predictably suboptimal models. So how to make this model selection faster was our goal. So this is what we did. We can now, based on the BIC and AICC scores, which is what we use to evaluate model fit, many of you know, we can predict what they would be, and we can skip those models that will have worse scores. So that's one way to save time. So here in Mega 12's new version that is available online, you have an option. You can choose filtered, which means skipping models, or full, which is slower, tries all the models. So you can skip models that are not going to be better. And here, a BIC of threshold 5, which is basically relatively small threshold. So it is very conservative in that it will not reject models too easily. That is used, but you can, this is the default option. You can change it to whatever you like based on your knowledge and interest. But essentially, we have now made available this filtered or fast model selection for DNA and amino acid sequences. So this is one example of 240 data sets analyzed in which Jux Cantor to GTR plus gamma plus I models were the real models, correct models, or the models that are obtained by using full slow process. And you can see if the model that we have is complex, like GTR plus gamma plus I, then that model actually you can save more than 70%, around 70% of the time. So basically what used to take maybe 2 hours or 1 hour or 20 minutes, whatever it is, now will take half as much or less. So that is saved and you get the same model. So ultimately, we did not lose on accuracy. Once in a while, you get a different model, but those models are equally good. So there isn't really a good reason to select one or the other. So that works in our calculations quite well. And this heuristic is very useful. In fact, for an amino acid sequence data set, which is a chloroplast sequence alignment, you could save 86% of the time. So this becomes 4 to 5 times faster. And again, selects the same model. So this kind of an approach is very effective in reducing the amount of time taken. So that's really what we are striving for in the case of model selection. The other thing that many people do that is very time consuming is bootstrap analysis. Of course, we also build phylogeny, but many times researchers develop bootstrap consensus trees. So Bootstrap is used for testing the tree as well as to infer a tree at the same time, which is originally what Felsenstein in 1985 did. And so we wanted to speed up Bootstrap too. And just to give you a quick sense of what Bootstrap analysis does for those of you who haven't done this before or haven't thought about it too much, since it is a straightforward process, you have the full data set. And from this data set, we generate a replicate data set by randomly sampling sites uh, from the full data set continuously. And essentially, some sites will be more than once in the resample, and some sites will never be picked by chance alone. And so the new data set has the same number of sites as the original data set. And then we do likelihood analysis, for example, or neighbor joining, or any other idea that one has, and we get a phylogeny. We can do another resample, phylogeny, another resample, phylogeny. We can do let's say 100 resamples or 1,000 resamples, get 1,000 phylogenies. And then we take all these phylogenies and we build a consensus, for example. And when you build a consensus, you get a tree like this. And in this tree, 99, 100, 94, these are what we call bootstrap confidence levels or bootstrap values. And they kind of indicate how strongly does the tree behave in the bootstrap analysis. So this is considered support for the hypothesis that is generated by bootstrap consensus tree. So now how everybody wants to compute these, and bootstrap is used really in a standard way in almost all the phylogenetic applications these days. So in Mega, we wanted to make this bootstrap efficient. Because what happens is when you go into doing bootstrapping, you get this option, choose the number of replicates to how many trees do you really want to make consensus. And there's no guidance. The old guidance from a colleague of ours was do 2,000 replicates. Well, 2,000 replicates takes 2,000 the time of building a likelihood tree, which is much, much longer. A neighbor joining might be easy to do for that, but to do a likelihood to 2,000 times is too hard. So people chose arbitrarily 100, 500, and then sort of grappled with it. So we thought, well, why don't we just automatically stop the number of replicates? We don't make you select some arbitrary number, but we do some sensible stopping. Keep bootstrapping until you reach a point where accuracy of the bootstrap value themselves is stable or good. So what we did is we now provide you what we call adaptive bootstrap. Adaptive bootstrap basically keeps track of how the bootstrap value is changing with time as you do more and more replicates. And once it stabilizes, then we stop bootstrapping. So in that case, automatically it finds the replicates. And so essentially the stopping rule is 5% error. That means bootstrap value is expected to have plus minus 5% standard error that you get. So the bootstrap value could be, if you get a 95%, it could be between 90 to 100%. And that is kind of okay for most of the analysis until the final tree that you want to publish, in which you can use more bootstrap replicates. So as you can see on the right side, Bootstrap with 500 replicates and Bootstrap Adaptive have a one-to-one relationship with minor variation as expected. So this works actually quite well. And we use some of the properties of Bootstrap value, which is just a binomial variance, to achieve this. But very interesting, if your data set is very strong, that means it has great phylogenetic signal for all the nodes in the tree, then that is this situation where all the nodes are 100%, 70%, 80%, or you have long sequences, irrespective of the nodes, bootstrap value sometimes. But if you have high bootstrap support, then you don't need more than 25, 30 replicates. So this saves a lot of time. But if your tree is very unstable, that means short sequences, short and many sequences, then you need more replicates. But still, you only need 125 replicates in our calculation. So you can get within 100 replicates or so, a 5% error, bootstrap values that are within plus minus 5% one standard error. So that's not bad at all. So essentially, Mega can now do this automatically. So this is basically 5 times faster in general. So in Mega 12, when you are given the bootstrap value using this approach and any approach you do, you get a range of bootstrap value. Well, this number is somewhere between 65 to 75. It could actually be 60 to 80 with small probability. But this gives you a good sense of where your tree is. And actually, for the same number of replicates, very high bootstrap values are actually estimated very with much more accuracy. So 95 to 99%, for example. So when your tree has stable branches, you don't need so many replicates, and you can achieve high accuracy of the bootstrap value. So that kind of gives you an idea that you can quickly uh, get bootstrap support you wish to get, you wish to have with respect to the stability with the Mega 12 application. Then in Mega 12, we have updated this, which is relaxed clock dating, but it's been available in the previous versions of Mega, which is an approach to estimate divergence times between species and divergence times between uh, taxa, sequences, genes. And this is one of those things that many, many people do, thousands of studies every year, estimate time for various reasons. And in 2000s, early 2000s, the sophisticated methods like Bayesian methods and others became available. They are really good. However, they have a number of assumptions, but most importantly, they are very time consuming. And we have been thinking, along with my collaborator, Koichiro Tamura, who is the co-author of Mega, how to make molecular dating really fast and not require so much time, and of course be appropriate. So we developed the real-time approach almost more than 10 years ago. And the real-time approach is very fast because, and show you why, but it's the fastest Bayesian method because comparison, this can be hundreds to thousands of times faster. So you can compute real-time analysis for a data set, almost within a few minutes, hours, rather than days and weeks it takes to do Bayesian, because Bayesian is much more involved. But they are very distinct approaches. And real-time also performs well in comparison to Bayesian. It can produce practically the same results as Bayesian method under same conditions. So once in a while it will disagree with Bayesian, but there's a good reason for that. And in that case, we do not know who is correct. So actually, real-time we found that since it was working so well, my colleague Koichiro Tamura and I, we sought a theoretical foundation for why this method works. And I should tell you that in real time, basically it uses branch lengths, which are likelihood branch lengths, and develops a set of equations, which are equations of relative rates. The relative rate between two branches is equal to the division of two branch lengths. You see R1 over R2 divided is equal to B1 over B2. And so this way, and assuming that the ancestral rate is an average of two descendant rates, because that is the best solution without any knowledge, it doesn't assume molecular clock because it allows the rates to vary across the tree and it allows the rates to evolve naturally. It doesn't use an autocorrelation model or an independent rates model or local clock model. It just, depending on where you are in the tree, it smooths out the rate variation, of course. And what we found is that we could estimate relative rates of evolution on all the branches using these equations as simple function of branch lengths. So for those of you who follow algebra and math a lot, this is like surprising that this is so simple. And it took us a few years to come to this realization because the initial method was actually an algorithm published in 2012. So in 2018, we published actual proof of the foundation of real time. So this really makes everything instantaneous. When you have something algebraic with equations and you have branch lengths, it takes zero time practically. So we have actually developed real-time framework extensively. We have now made it such that it can do node dating, it can do incorporate fossil calibrations, it can generate truly correct confidence intervals that are very difficult to do in many other approaches, like 3PL or other faster approaches. It can be used to develop the test of autocorrelation. So a lot of these features are all available in Mega now, in Mega 12 and previous versions of Mega 2. So this is another really fast approach. So as you saw, I showed you three different types of major areas that people analyze, various types of questions they have about dating, about building trees, about some model selection. And Mega now excels in all three of them in achieving much greener computing. So now I'm going to shift focus for the next uh, 15 minutes and I'll talk about future additions to Mega, which is what I want to do in Mega when you have data sets that contains thousands of tens of thousands or millions of sites in the alignment, which is phylogenomics, very long sequence alignments from transcriptomic data, from genomic data, from exon data, exome data. How do you do that? Well, many people will say, hey, let's just use the supercomputer. But not everybody has supercomputer, as we'll talk about. So we like to do this on a desktop and I want to do this within our lifetime. That means it should be done in the next few hours or minutes or whatever. So this is what I'm interested in. So I'm going to start with the bootstrap analysis, which is the most time consuming. Uh, so sequence data has grown immensely, as this is just a simple picture to show you that this is really exponential increase very recently. And this is just the number of bases in the GenBank, one simple place. For those of you who may not realize, I finished my PhD in 1996, and then the data was minuscule compared to today, and life was much simpler. Now life is more exciting and more challenging. So that's where we are today. And big data are now common in phylogenomics. Everywhere you go, people have millions of sites, thousands of sites, hundreds of thousands of sites for many sequences, sometimes hundreds of sequences, many times 30, 40, 50, but a number of times a couple of hundred or even 1,000 now. So computational cost of phylogenomics when you do bootstrapping is, as you can see with the red curve, increases fast. It may take, for a data set of 400, 500 kilobases, uh, 50 hours to compute just for building a tree. Memory can be even bigger problem for desktop, 25 gigs of RAM, for example. So you can't really fit it. So long sequence phylogenetics or phylogenomics is very problematic. It requires lots of memory, excluding a lot of people who don't have these fancy computers from research. Requires hours and days of computing. This is very problematic, uh, particularly in countries where you don't have a guaranteed electricity running every day or every hour even. Also, for people who have all of these facilities, it reduces rigor. They can try different models and different methods and different exclude or include genes to see what the effect is. You really can't do everything because if you have to wait for many days for one calculation to finish. And ultimately, if a paper is published, uh, it is very difficult for anyone else to reproduce what the result is because you need a big computer, you need a lot of time, and you need somebody with an ability to run those analyses. So this has resulted, in my opinion, a lot of lack of testing whether the actual published results are as strong or as weak as they are published to be. So this is a problem. So a lot of this is not green as well as not scientifically rigorous and reproducible. So we introduce this idea of little bootstraps. So little bootstrap is like regular bootstrap, except for a major change that we have the full data set. And instead of sampling all the sites with replacement to build a complete alignment, we actually sample, let's say, 1% of a million sites randomly. And we make one little data set. And then we take the little data set, and now from this, we build a full data set. And this full data set is actually replicate, similar to Bootstrap, regular Bootstrap, but this time it contains fewer patterns because original data contained, let's say, 500,000 patterns of site configurations. and this may contain only 10,000. So this means that if you do ML analysis of this data set, you can do it very quickly. Like essentially, if the subsample contains 1% of the site partitions or configurations, likelihood method will be done in 1% of the time, so 100 times faster. And compared to one tree. And then you just keep doing multiple upsamples, they're called, and you build lots of likelihood trees, or neighbor joining trees for that matter, and you get a bootstrap value for a given node that you're interested in. Then you do the same thing and do another subsample, do a bunch of upsamples, another subsample, bunch of upsamples, and you get the bootstrap. So basically, for any node you are interested in a tree that you may have a priori, you could compute bootstrap support value from each subsample. So this is basically random samples of data that are then upsampled and bootstrapped. And then you take an overall aggregate. So what little bootstrap is like this. Felsenstein in 1985 introduced Efron's bootstrap approach to molecular phylogenetics, and it has been revolutionary. However, it becomes very slow for very big data sets. So what we did is we introduced the bag of little bootstraps approach that is proposed by Kleiner and Jordan in their 2014 paper, and made some updates to it so that it can be applied to our field, and published it. And hopefully, I mean, in my case, I'll show you in a moment, it really makes bootstrapping huge alignments very fast. So this is something we want to add to Mega in the future, but right now you can analyze it using the GitHub repository. There is software there to use for it. So look at the little bootstrap. Can you find where little bootstrap is and time taken? There. So little bootstrap took only few percent of the time of the full bootstrap. So that's really fast. And little bootstrap also took a very small amount of memory. What took gigabytes of RAM now takes megabytes of RAM. So essentially now you can analyze this big phylogenies on your desktop. And we, in our paper, if you are interested in that, found a way to automatically determine the number of sites to subsample, so you don't have to choose that arbitrarily, number of subsamples to do, so you don't have to do it yourself, and the number of little replicates per subsample. So with these three advancements, we made little bootstraps adaptive and essentially give you a result that is very similar. So if a node is supported at 95% level in completely fully bootstrapped analysis standard way, then in almost all cases except one or two nodes in the tree, little bootstrap generates 95% or higher support. This is what this graph shows. And actually, in the cases where it fails to do that for some nodes, we have some work now that shows that those nodes are actually extremely problematic for concatenation analysis, and they may in fact be spurious. So essentially, this is the work we are doing, and we'll talk about that hopefully next time. But the accuracy of little bootstrap is really the same as the accuracy of is very high. That means it's the same result as the full bootstrap. And so with that, we solve one problem. But before you can do bootstrap, you need to choose a model. So if you have a data set that is huge, model selection can take forever. So we had to also, in order to speed up the whole pipeline, we had to also go and do model selection speed up. So we actually were able to adapt the little bootstrap subsampling and upsampling approach, which is published, and be able to show that for model selection, it can actually speed up even more. So other than taking 1,000 hours for some data sets, very big data sets, it only took few hours. So it had 200 times speed up. So speed up is a lot more than the speed up for bootstrapping. And the memory taken is also very small, from tens to hundreds of megs rather than 100 gigs in this case. So suddenly, using this subsampling and upsampling approach, we are able to do these two processes on a desktop very quickly. So why does it work? So what happens is that unlike traditional subsampling approaches, the idea of upsampling introduced by Kleiner and Jordan that we brought into the field and with some modifications, it actually restores the power of the data analysis. And in fact, the amount of evolutionary substitution in the upsample data set, which is the SU data set, subsample upsample data set, is proportional to the full data set as compared to subsample only that doesn't contain much. So even though the computing time is short, the number of substitutions is similar to the full data set. And it's because we upsampled a small sample. And very interesting thing we found was that all these substitution rate parameters estimated using this model Taylor approach that I just talked about, subsampling, upsampling, are virtually identical to what you get by full analysis. So you really don't lose any accuracy. So again, no accuracy loss, but still huge benefit. So what I wanted to conclude in this portion was greener computing for big and small data sets. We have now made many developments that are going to make big data analytics on our desktop computers much more feasible. So we have model tamer for optimal model selection, little bootstraps for phylogeny inference, automated selection of subsample size, number of subsamples, number of replicates for all of these methods. We have real time. We have heuristics for faster computing, and we have greener computing, which is classical methods are made green, but also we have started to do machine learning, which can sometimes be much faster than classical methods for certain applications. So one of those machine learning approaches, which is based on evolutionary sparse learning, is in Mega 12. In fact, if you have data set which contains some contaminant genes or sequences, for example, in this data set, there is a big green dot for a node where you can ask the question, does this node correspond to a place where the analysis or the inference is disrupted by a problematic sequence in some one gene, maybe in one species or whatever. So you can actually run this machine learning approach in Mega 12. It takes sometimes it can take a half an hour or 15, 20 minutes because it is analyzing large amount of data. It can take thousands of sequences, thousands of genes, I mean. And it will produce an output like this. It on the x-axis is the species name, and on the y-axis is the name of the gene. So whenever there is a color other than green, that indicates that a particular gene in that species is problematic. Now, this one doesn't require any likelihood analysis. It does evolutionary sparse learning. I won't go into explaining this, which can take a little bit of time. But in Mega 12 now, you can do this. You can actually diagnose problematic cases, and you can see a few pink and orange colors there, which are all problematic genes. So and then when you have this gray means missing data. And green, dark green means the most influential gene. If you remove this gene, out of 1,000 genes that are used here, the tree may actually change. And this is the example in which it does change. So, but this you find automatically without having to build gene by gene trees or likelihood analysis or any of those things. And you can generate hypotheses of problems. So Ascoidia rubescens clearly in this case, the first species is problematic in some way, and it is indeed, as published in original studies. So anyway, so I'm going to go away from phylogenetics of inference and spend a few minutes talking about our work on biomedicine. And in biomedicine, we have done a number of research projects, but the one I'll talk about is the most interesting one in the recent couple of years for us. So in this case, we have done previously research in phylomedicine. Phylomedicine has to do with saying something about our genome variants that we carry using the knowledge of what nature has permitted in the past. So this is really all about germline type of mutations. in the case of animals. And essentially here, we use evolutionary tree and evolutionary conservation and divergence to inform which variants that we carry today, which are de novo or unique in some ways, they're detrimental. But that's something I will not talk about today because that's a little bit older topic in our lab. But instead, after the conception, as the cell divides into trillions of cells, which make up a human, mutations still happen. And these mutations are on a personal time scale. So these mutations sometimes can be quite large. And one of them is a high turnover situation where some cells are turned over a lot. For example, stem cells for blood. So these high turnover cells make us think that extending from conception to somatic variation is a Biomedicine 2.0. I'm supposed to write a review article about that perspective, but I have not had a chance to. But that's how I think about it, that from the first Biomedicine 1.0 described in one of my previous works, in which a lot of people have done work in, to this Biomedicine 2.0 about blood and other high turnover tissues, just such as intestinal cells and others. That's what I'll talk about. So here is primarily my discussion today is about hematopoietic stem cells. And hematopoietic stem cells actually are progenitor of 10 plus types of blood cells. These lineages are established early on. By the time we are born, we essentially have huge number of founder or embryonic stem cells, which are formed. And these stem cells then give rise to blood cells. And of course, because these stem cells have to be replenished continuously, and they divide through asymmetric cell division, actually, they accumulate mutations, obviously. With every cell division, there is a chance to accumulate mutations. So suddenly, thousands of cells, hundreds of thousands of cells accumulating variants, there's definitely a phylogeny of these variants. And so it's a variation that is somatic variation in our stem cells. So there was a very cool study by Mitchell et al. A couple of years ago, in which they actually did sequencing of 10 individuals. And in fact, did single cell sequencing using cell sorting, clonal expansion of cells, and whole genome sequencing. This way they could get really highly accurate single cell sequences, which is not possible from direct cell single cell sequencing. And what they showed was something that is known in the field for a while, but they showed really beautifully that with age, the mutational burden that is accumulated in stem cells is actually increasing almost linearly, like 17 to 18, 14 to 18 variants per genome per year. So this is incredible molecular clock, almost like a perfect molecular clock. But in another back-to-back paper, Williams et al., what they did is they did the same thing in patients that suffer from myeloproliferative neoplasms. And now they were also able to estimate the mutation variants that are accumulating in these patients' stem cells. And this is the picture they show, which is kind of linear. I mean, kind of very linear, I should say. But actually, if you superimpose the healthy on it, it's almost the same. So really, there aren't too many additional mutations accumulating in patients with MPN. They are accumulating at the same rate as healthy individuals. So that doesn't really explain why these individuals got MPN. Of course, they may have some driver changes in cancer that caused it. But in fact, in the left side, you see, is the incidence of MPN. Incidence of MPN increases exponentially with age. Everybody's okay. We are all okay until 50s. But then after we go past 50s, it really just takes off. Exponential increase. But that exponential increase is not explained by this linear change. So we analyze this genomic data ourselves just to be sure that we get similar results or not. And you can see MPN incidence per 100,000 people in red and their single nucleotide alteration count, they don't jive with each other. They're very different. One exponential, one linear. So one thing we noticed, because we are phylogenetics people, we are interested in phylogenetics in everything, that if you look at the phylogeny of cells in the patients that were sequenced by healthy patient, in the individuals, healthy individuals sequenced by Mitchell, the infants have a phylogeny like what you see on the left side, with a few hundred HSCs that they sampled. So that is sort of very short time. So this is shown here. But in a 20-year-old individual, there were many changes. Then in a 48-year-old, there are more changes. So you can see these long branches, which is because the variation, the cell division is asymmetrical, so there's not many bifurcations. So 63, these branches become longer and longer. And by 77, suddenly, not only the branches become longer, but also white space is created in these phylogenies. And white space is corresponding to extinction of certain embryonic lineages and formation of secondary lineages that are coalescing, let's say, recently in time. And by 81, the tree become even more, it has a lot more white space, which means more ancestral lineages are being lost. And so phylogeny shape is aging. So we took inspiration from this observation that were made using Mitchell's data and thought that we could track the phylogeny shape in as a measurement of risk. So we developed a few new approaches, methods to actually estimate this diversity decay over time in healthy individuals. And when we made this diversity decay and did a correlation with age, they were really amazing fit. So essentially, the phylogenetic diversity decay can actually be used to generate a graph very similar to the risk of MPN or cancer, blood cancer. So this was very interesting and very exciting. So we used this to build a model, very simple logistic model. And we were able to build this model and predict chronological age based on genetic trees or phylogenetic trees. And we got good results. But when we applied this model to MPN patients, for a patient of age 49, their age in phylogeny age, we call it phylo age for HSC or blood health, was 94. So almost twice as old. And actually, this is hugely greater risk of cancer. So this phylogeny is even worse than a healthy 81-year-old, which is exactly what our phylo age concept tells us. So essentially, we found a way to quantify the phylogeny and convert this or translate it into the blood age, blood HSC age in the phylogeny terms. So their increase was almost 50 times more likely to develop cancer if one has an HSC phylogeny similar to the one above, which is to us was astounding. It's possible. Now, a lot of people are interested in physiological aging of tissues, and this is one, I think, a new way to do that. Then we did this for all the patients, and we found that all the patients' phylo ages were much older. Like a 20-year-old patient, phylo age was 60, and a 60-year-old phylo age was 85. So essentially, most people had phylo ages with cancer older than their actual age. And recently, we have been developing phylo ages from targeted sequencing data, which, because single cell sequencing is very difficult, particularly the colony type sequencing. And we have a new article that is currently in review and revision for MBE, which we applied to the TCGA AML cohort data and found that just like the analysis I just showed you from colony single cell sequencing, all the patients were actually much older than their chronological age. So this is exciting that we could potentially make it translational in research laboratories, which can actually do not really bulk sequencing, I would say high quality bulk sequencing like 300x or targeted sequencing of many genes or the exome. And so I'm getting close to ending the presentation. So I just talked about high turnover, but there are also phylogenies of tumors that are not high turnover, but rather these are benign and malignant tumors that are more solid or much more of a mass. And so in this case, this is also part of Biomedicine 2.0. We have developed many approaches for estimating, for making single cell sequence data better through imputation, which is evolutionarily inspired, finding clones in the bulk sequencing data, looking for patterns of migration of metastases in a body uh, retrospectively, and also detecting shifts of mutational signature over time. But these are all phylogenies informs medicine uh, kind of research we've done in the past few years. And then just to sort of wrap up, for those of you who don't know, we have a database of time trees of life. I'm coming back to my phylogenetics roots here, and where you can find out divergence time between any two pair of species for which we have data, which is 140,000 now. You can put in two species names or common names, and you can get their divergence time along with how many studies are published. It on the left, you see lots of black dots. What is the variation in people's thinking and the literature? Here it will give you 65 different studies that have published this divergence time between human and mouse. So this is a data set. This is a database of results where you can get the results quickly, especially if you're not from the field and don't understand the jargon in molecular systematics and taxonomy. So I want to conclude by saying that molecular data-driven discovery is now at center stage in most areas of biology and medicine. As data scholars and scientists, my group and many groups, in fact, are identifying emerging needs constantly and developing new methods. We are very interested in end-to-end practical solutions, and our emphasis is on greener computing for better environment and for broader participation. I want to acknowledge my colleagues at Penn State, ASU, and Temple University over the last 30 plus years, long-term collaborators, Koichiro Tamura on Mega, Blair Hedges on Time Tree, for example, and a number of team members without which none of this would have been possible. You can have some ideas, but you really need amazing people to execute it alongside you, or you do that alongside them. A lot of funding agencies supported this work over time. Thank you so much for your attention.